So students, antigens are those compounds which stimulate the production of antibodies in the human body, and antigen can be, remember, antigen can be self-produced. That means antigen can be self, or otherwise it can be non-self. And in the same manner, the antigen can be complete antigen, which is capable of inducing antibody formation, and then incomplete antigen, which is incapable to do so but is able to do if it is combined with some carrier protein. We learned that the antigens are basically proteins, but there are some antigens which are even carbohydrates, lipids, as well as nucleic acid. So the foreign molecules that have the ability to bind with the components of the immune system are called as antigen. I even explained in the last classes to you that the antigen, complete antigen is not important. It is a particular part of antigen which is called as epitope. So epitope is the one which attaches with the antibody or attaches with the receptors which are present on the cell surfaces of B cells and T cells, and then it will stimulate. That means epitopes will stimulate the immune system to start producing antibodies or to start performing the proliferation of the effector cells or the plasma cells. So let's start with the topic of antibodies. We understood that antibodies are also called as immunoglobulins because they are the proteins which are involved in providing immunity to us. They are produced by the actively involvement of immune system of the body, and then antibodies specifically inactivates a particular antigen, and antibodies belong to the gamma type of globulin proteins which are present in the blood serum or blood plasma. Then there are types of antibodies which we are going to learn today, and antibodies constitute the humoral immune response or humoral immunity. So cell-mediated immunity is governed by T cells, and B cells which release antibodies are a part of humoral immune response. Now we have already did the structure of the immunoglobulin, that it is Y-shaped. I found many students making wrong diagrams because they never drew these disulfide bonds properly. Please try to draw and practice these diagrams a number of times, because in biology, if you are considering full marks, then it is possible only if you are mastered with your diagrams also, plus labeling of the diagrams. So we learned in the last class that the antibodies are made up of 2 heavy chains and 2 light chains. That's why it is generally represented as H2 and L2. So 2 heavy chains and 2 light chains are present in this particular specific manner. This portion, the lined one, this portion is the constant one. And this is the variable part, and this variable part is antigen binding portion of it. This is the constant part, which was first discovered as a crystallizable part, so it is also called as crystallizable fragment. This is the fragment which joins with the antigen. That's why they are named as FC and FAB. This is for a specific antigen to bind with. And remember, this binding is going to be a lock and key type of binding. So a particular antigen only can fix inside this antigen binding protein portion of the antibodies. Mature B cells, plasma cells, are able to synthesize at a very high rate the antibodies, and that rate can go up to 2000 molecules of antibodies per second. And remember, this is a part of humoral immune response, or it is a part of antibody mediated immune system. Let's understand the differences between antibody and antigen. Antibodies are proteins, but antigens can be proteins. Sometimes they can even be lipids or nucleic acid or otherwise carbohydrate. But majority of the antigens are also proteins. Antibodies are synthesized by the animal's body or human's body to combat or to fight against the foreign material. And antigens are those foreign materials which will cause antibody to be formed in the animal's body. Antibodies occur on the surface of the plasma cells, that means B cells, as well as they are present in the body fluid. That's why it is called humoral immune response. And antigen are present on the surface of the microbes, and they can also be found as free molecules. Antibodies join to the antigen to cause destruction of the antigen, and then antigen binds to the macrophages to reach the helper T cells to initiate the immune response. Now let's understand the various types of immunoglobulins or the antibodies we have in our topics for your NEET exam. So the antibodies are of various types, and remember, antibodies show following characteristics. So I have written their short form, their percentages, where are they present and how do they function. It's all into a small summary type of. So when we talk about IgG, the first one I have mentioned is IgG. So IgG is the most abundant class of immunoglobulins in the body and constitute approximately 80% of total number of immunoglobulins. It is found in the blood, it is found in the lymph, it is found in the intestine, and remember, it is able to cross the placenta. So it is placenta permeable type of immunoglobulin. No other antibody is able to cross the placenta, only IgG is able to cross the placenta. It provides protection against the bacterial infection, viral infection, and by enhancing phagocytosis. It is the only class of the antibody which is able to cross the placenta from the mother side and reach up to the fetus. That's why it is able to provide, it is a part of passive, natural passive immunity. We have already covered this in the previous class. The next category, one more thing you need to remember is IgG is more capable of fighting viral infections compared to the bacterial infections. Then the next one is IgM. The IgM, from M you can make out, M means macro. So it is the largest of all the immunoglobulins.